Okay, hi everyone. Very good morning. So basically, this is our second lecture. Okay, so we will continue on our second lecture on the sampling storage, sampling, see here, sampling, storage, and sample preservation. Okay, so before this, we have learned about the general steps in chemical analysis. So for this second lecture, we will continue with sampling, storage, and sample preservation. Okay, basically, what is a sampling? Sampling, it is a process to get a representative and homogeneous sample. Okay, so basically now the important thing is the word representative as well as, representative as well as homogeneous. Okay, representative means that the content of the sample reflect the bulk of the sample. Okay, reflects the bulk. Means that apa yang kita ambil itu representative of the whole bulk yang kita nak test tu. So for example, if in the case of apa in the case of for example a fish that has been dead, so we want to know on what is the cause of death, whether it's been intoxicated ke, whether it's low oxygen level ke. So means that the sample that you take, the sample that you want to sample must be representative. For example, you take the blood. So the blood is representative because it flows throughout the body of the fish. Okay. It is also must be homogeneous, means that the sample must have a same content throughout. Okay, so for example, if you were taking the sample near the stomach, which is much more better because everything that has been ingested, the content is going to be more or less the same throughout that area. That's how you get a homogeneous sample. All right. So, deciding on how to obtain a sample depend on the size of the bulk, the physical state, and the chemistry of the material to be assayed. Okay. So basically, what are the steps that being involved in sampling a bulk material? First of all, you need to identify the population from which the sample has been obtained. You need to collect a gross sample, and then you need to reduce the gross sample to become a laboratory sample that is suitable for analysis. Okay, so basically, these are the general steps in sampling a bulk material. Okay. All right, the next slide. We are going to look at on how are we going to sample a solid. Okay. Uh, Inhomogeneity of the material making sampling of solids making the sampling of solids much more difficult. Okay, as kan contoh lah in this case for example, in this guni, okay, in this guni, the sample is not basically it is not homogeneous throughout the guni. Okay, maybe there are some things that are much more high in concentration towards the left side or maybe towards the right side. It depends on the sample. It also depends on the bulk. So basically, when we are sampling solids, the easiest way to sample solid is by using a grab sample. The sample is taken at random and assumed to be representative. Okay, we assume that what the places that we try to get the sample or the solid sample is representative. Okay, how are we going to do this usually? Okay, for example, in this guni kan, in this picture, so it is a rectangular shape. So usually what we do is that we will take maybe one from here, four sides, and one in the middle. So basically we have 5 samples that we will say that it is a taken at random and assume that it is to be representative. Okay, for reliable results, usually we will take 1 over 50 to 1 over 100 of the total bulk. The larger the particle size, the larger the gross sample should be. So 1 over 50 of the total bulk. Okay, for example, if the bulk is about 50 kilograms, so 1 over 50, so you should have a sample about 1 kilogram sample. Okay, so for example, you can see here the stockpile of grains taking increment from surface and interior via grab method and the motion method. Okay, this is not that important, but the the most important thing is that you need to know on how to sample a solid. It doesn't mean that this is the only sample, but other sample also can be using the same principle, if I may say. Okay, so we'll continue on the next slide. So basically, obtaining a random sample from a bulky material, for example, as I said, ore, grain, or coal, can be achieved while the material is in motion. Periodically transfer the portion into a sample container. So periodically ni maksudnya it says that okay, it is in conveyor belt, so you take randomly. Okay, you take something at the front, something in the middle, and some at the back. Okay, you put into a sample container. So basically that is called a gross sample. All right, so the gross sample must be reduced in size to obtain a laboratory sample. Okay, how do we using that? It is called a cone and quartering method. Okay, so the sample, the gross sample that has been obtained, we crushed and mixed to form a conical pile. This first one. The pile is then flatten this one, the second picture, and cut into equal quarters. And two opposite quarters are chosen at random. The third one and the fourth one. Until the gross sample is small enough to be transported to the laboratory. Okay. So as I mentioned before, for example, if you have 1 kilogram of sample, then it would not be feasible to carry all 1 kilogram back to the lab. So this is how you're going to reduce the gross sample to become a laboratory sample. Okay. All right. How are we going to sample liquids? So basically now, I think that you can see the pattern here. When we are sampling, there are sampling of solids, sampling of liquids, and then sampling of gases. All right, sampling of liquids. Liquids are much more homogeneous as compared to solid. All right, the gross sample can be relatively small. If the sample are not homogeneous and have small quantity, they can be shaken and sampled separately. Sampled immediately. If it is not, contohnya if you take some river water, sometimes at the bottom part of the sample, you have some sort of like a sediment. So usually for this kind of sample, you just shake it and then we have a much more homogeneous sample. All right. Sampling techniques will depend on the types of fluid. So usually we will be using this kind of, if you can see here, this is called a sample teeth. Sample teeth. Okay, so this sample teeth, it is in cylindrical punya shape where you can sample your liquids. Okay, so how does this happens? So basically it goes without saying, when you open the opening, then the liquid sample will be flowing into the sample tip, and then you close it and then you just take it out and bring it to the lab. Okay, so examples of liquid samples, for example, large volume of liquids, um, large stationary liquids, and then biological fluids also can be used. But biological fluids usually kita tak guna sample tiff lah. We will be using macam syringes ke ataupun a small collector. Okay? Alright. Alright. So, how are we going to sample gas? Okay. Gas is also tends to be homogeneous. All right, large volume of samples is required because of their low density. So if we were to look at on how much is the sample needed for each of the following, gas is going to be the highest volume because of their low density. So usually, for example, in the case of Pasir Gudang hari tu, usually we'll be using like a high vol sampler where we just place it there and then it will just straight away collect all of the sample or all of the atmospheric sample and then it will be placed onto an absorbent. All right, so this is the usage of high vol sampler, and then the gas or smoke from burning activities. Usually we'll be using a gas sampling bulb glass, and then for example the most simplest thing is the breath sample. You punya pernafasan you punya gas tu. So we will be using into a sampling bag. All right. So basically, you have learned on on what is sampling, and then how to sample liquids, how to sample solids, and how to sample gases. All right. So now we are going to sample storage and preservation. All right. So basically, the objective of sample storage and preservation it is to preserve the nature of the analytes in the sample as similar as possible during the sampling process. We don't want the nature of the sample to be changed from time to time as we are waiting for it to be analyzed. So that's why we memang penekanan diberi terhadap sample storage and preservation. Alright, so sample container materials can introduce either positive or negative errors in measurements, particularly ultra trace levels. Okay, okay. For example, ultra trace levels ni, contohlah you sample liquids dalam sample TIFF tu kan, sample TIFF. Okay, you nak measure apa? Heavy metals inside the liquids. But some heavy metals have the ability to latch itself, dia melekatkan diri dia kepada sample container ini. So bila dia melekatkan diri kepada sample container, then you punya apa orang kata tu, you punya concentration, intended concentration yang you nak cari itu, it will deviate. Okay, sometimes it will have a positive deviation, sometimes it will have negative deviations. Alright, so usually what do we do in terms of preserving, in terms of avoiding these things to happen? So the mechanism of preservation is it is usually, for example, refrigerated or freeze. Okay, we freeze the sample after the collection until the time of analysis. This is usually done for biological samples through the action of bacteria, because bacteria would have the ability to degrade your biological samples. And then the second way is usually we acidify it with 10% of nitric acid immediately upon collections. This is to prevent the precipitation of metals from water sample. When it is when your heavy metals have precipitate, means that your sample is not homogeneous anymore. So we want your sample to be homogeneous. As what we have learned before, we need it to be homogeneous. Okay, we need it to be representative. If it had the heavy metals have been precipitate, then your sample is not homogeneous anymore. Okay, so these are examples. For example, kalau untuk hydrocarbon samples, we will using these metal containers, amber vials and bottles ni usually for light sensitive samples. Ada certain samples that is very sensitive to light. It tends to oxidize by the presence of light. So we need to use the amber vials or bottles. Okay, polyethylene bottle also can be used for organic trace analysis. All right. So, okay. So basically, what I want you to do is that for this particular slides, okay, I want you to suggest to me. Okay, let's look at this question first. Okay, how do we sample based on the three pictures? These three pictures. The first picture, this is the first picture, the second picture, and the third picture. Okay, how are you planning to sample these three situations? How do we sample based on the three pictures, sea, soil, and soot from the Gulf of Mexico disaster? Also discuss on how to store and preserve. All right, so for this activity, I would need you to so this is going to be an individual activity. Okay, so I will have a link inside the e-learning for you to submit. So this activity, this submission will be counted as your attendance for this asynchronous lecture. Okay, so basically just jot down on whatever piece of paper that you have on how are you going to sample these three pictures. Okay, the sea sample, the soot sample, as well as the soil sample. And then how would you store and preserve it? So the analytes, it depends on you. It is up to your imagination. But I want you to have some idea of this on how are you going to sample these three type of samples, and then how are you going to store and preserve it. You are free to Google as well. Everything that is been logical, it is accepted. Okay, so all right. So for this activity, jot down on piece of paper, take a picture, and upload it into the e-learning. All right. So this activity, this submission will be counted as your attendance for this asynchronous lecture. All right.